Morning everybody, welcome to another edition of the Lab 207 webcast. My name is Mr. Kite. I'll be hanging out with you today. And as you can see, we got a new template, so that means that we are starting a new series. This series is going to be about cell division. There will be several videos in it on several different topics. Today's topic of concern is the cell cycle. So like always, let's go ahead and get our objectives, and then we will get on into the meat of our lesson today. So by the end of the video, I need you to be able to know or do the following things. Identify the parts of a chromosome. Second thing is to explain what happens in each stage of the cell cycle. And finally, be able to describe each of the steps of cell division. A lot of stuff to cover today, so let's get into it. Anytime we're talking about science, vocabulary is super important. We need a basis for which to be able to talk about our topic. So here's some important words that you will need to know as we work our way through this series. First one is the genome. Genome is just a collection of all of the genetic material that makes one organism unique from another. All of your genes in your body make up your genome. All the genes in your dog's body makes up the genome of your dog. Chromatin is a strand of DNA that has been wound around some proteins. DNA is a hugely long but terribly thin molecule, so it needs some organization in order to keep it from getting tangled and broken or whatever. So take a DNA strand, wind it around some proteins called histones, and you have got chromatin. Take that chromatin and further wind it up into a tightly condensed coil, and you have a chromosome. Know that one chromosome is one strand of DNA wound up into that nice little X-shaped pattern that you are so familiar with. Next term to note is a somatic cell. These are all of the cells in your body except for eggs and sperm. Know that somatic cells have all got 46 chromosomes, 23 from your mom, 23 from your dad. And while we're talking about chromosomes, let's talk about sex cells, also known as gametes. These are eggs or sperm, and they're the only cells in your body that have got 23 chromosomes. Talking about chromosomes, let's get into the anatomy of a chromosome. There are three major parts of a chromosome that you need to be able to recognize very quickly. I'm going to point them out with my pen. So you're all familiar probably with the chromosome picture that looks like a nice little X like that. You can see this here. They're just laid over sideways. Major parts that you need to know. First one is the centromere. That is the center of our chromosome where everything is connected together. Second term that you need to know is a chromatid. One leg of our chromosome is known as a chromatid. The one that matches it on the other side is called a sister chromatid. So these two right here are sister chromatids. One of them is a single chromatid. Both together is a sister chromatid. If you can remember those terms, you should be pretty good for the rest of this section. But we need to talk about today is the cell cycle. The cell cycle essentially tracks the life of a cell all the way from the time that it divides until it comes around and divides again. The events of the cell's life are organized into this little diagram here called the cell cycle. So let's talk about a couple specific pieces of it. You can see right here, our brand new cell starts the cell cycle by entering the G1 phase. G1 is where the cell is essentially doubling its organelles, it's doubling its cytoplasm, it's gathering up more materials so that it can divide again. Most of the life of a cell is spent getting ready to divide. So this is growth 1 stage. As our little cell cruises through this growth 1 stage, he is duplicating organelles, his cytoplasm is growing, he's getting a little bit bigger. He then enters the S phase, which you can see right there is where DNA synthesis happens. If our cell is going to divide, he needs to make a new set of DNA because, if a cell was to divide without making a new set of DNA, that means that each cell that resulted would only have half the DNA of the original cell. Half the genome is a problem. So all of the chromosomes inside of that cell need to be duplicated so that the cell can divide and each new cell gets half of that chromosome or half that genetic material. So DNA is being synthesized through here. In G2, we are making final growth and preparations. More cytoplasm is growing. We've got organelles finishing duplicating. Cells being checked out before it enters into M phase, which includes mitosis and cytokinesis. We're going to talk about each one of those in more detail in a second, but know that this whole thing is the cell cycle. Also, another thing to note is that phases G1, S, and G2 are all part of interphase. I'll talk about interphase in a second, but each of these phases is considered to be part of interphase. Speaking of interphase, and moving on into the whole idea of mitosis, the thing I want you to know about mitosis is that it is like a Xerox machine or a copier. One cell enters mitosis, two identical cells come out the bottom. There is no difference between the two of them. They've got the exact same genetic material, which means that internally they are exactly alike. So just know one cell enters mitosis, two daughter cells that come out. Those daughter cells are exactly like the parent cell. We're going to go ahead and take a second to go through each of the stages of cell cycle and mitosis. So we're going to start out with interphase. Like I told you a second ago, interphase includes G1, S, and G2. This whole interphase is where the cell is making preparations to divide. As far as the life of a cell is concerned, it spends 90% of its life in interphase. So it spends most of its life getting ready to divide. Let's go ahead and talk about those individual steps of cell division, starting with prophase. Now, there's an acronym that I use to remember all of these stages. It is IPMAT with a C on the end. The I stands for interphase. We just talked about interphase. We are now on P, which is prophase. I think of prophase as organizing the chaos. So we've got our cell. When our cell is just hanging out in regular interphase, all of its genetic material, which is represented in blue right here, is all wound up and tangled up as chromatin, and that chromatin is like a big old mass of spaghetti. It would be really hard to divide this mass of spaghetti such that all the same genes ended up in the two daughter cells. So one of the things that happens in prophase is our mass of chromatin condenses into individual chromosomes. A couple other things that happen. The nuclear membrane starts to break down because if our cell is going to divide and those chromosomes are going to move, they can't be held in by the membrane. Last thing that happens is there is an organelle called the centromere. Centromeres send out spindle fibers, which you can see right here. These spindle fibers are going to go out and connect to each of the chromosomes. So a spindle fiber would come and connect to the centromere of a chromosome, and each side of the nucleus is going to have one of these centriole complexes called a centrosome, sending out spindle fibers to connect to the individual chromosomes. Next phase is metaphase. I like to remember metaphase military, and the reason that I remember metaphase military is in metaphase, all of our chromosomes line up across the middle of the cell. The place that they line up is called the metaphase plate. All right, that is essentially the middle of the cell. So the metaphase plate would be right here. It's not an actual physical plate or anything like that. It's just a line that our chromosomes line up along. And you can see very well in this picture right here, our centrosome is right here, and all of these spindle fibers are going out. Each spindle fiber is connected to one side of a chromosome at its centromere. So remember, metaphase military. Anaphase is the breakup. This is where our chromosomes actually start separating from each other. So if you remember, in that S phase, our chromosomes were duplicated. It's not until chromosomes are duplicated that they have this nice X shape. In this X shape, one side is identical to the other side. So all the genes that are on this side of the chromosome are also on that side of the chromosome, which is really important when we look at anaphase. In anaphase, all of our chromosomes are being torn in half, so you're taking your sides of your chromosome and going like that. If they were not duplicated, you'd have different genes going to different places. So if we have got half of chromosome A here, we have got half of chromosome A there. The spindle fibers are shortening, which is pulling our chromosomes, our chromatids, back towards each side of the cell. So in this phase, the sister chromatids are being separated from one another. Next we get to telophase. Once we get to telophase, we have got two distinct nuclei in the cell. If you look at our little micrograph here, all the blue are the chromosomes. There is one set of genetic information. Here's the other set of genetic information. At this point, each cell has got a full set of genetic information because we had done that duplication beforehand. The nuclear membrane is starting to reform, which is going to enclose all this genetic information. The cells, you can see, are starting to separate from one another, and we are pretty close to having two distinct cells. Final phase is going to be cytokinesis. This is where you get two cells that essentially have a life of their own. If you notice those blue blobs on the right there, our chromosomes have unwound. They are again a big mass of chromatin. Nuclear envelope has reformed to enclose that genetic material. One quick term to know is cleavage furrow. When our cell divides, a cleavage furrow forms, which is essentially this place where the cell pinches in. That's called the cleavage furrow. That area is going to just continue to pinch in like the drawstring on a pair of sweatpants until it pinches the two cells in half. Last slide, last topic for the day is kind of an outlier. When bacteria need to divide, they go through a process called binary fission. And the way that works is simply this. Bacteria have got a circular chromosome. They don't have all of the X-shaped chromosomes kind of like we do. They don't have nearly as much genetic material. What happens in binary fission is this circular chromosome essentially starts to get a loop in it. So if this had two sides, one side would loop down, and the inside of our chromosome is going to be duplicated, and these two loops are going to essentially pull apart from each other. They are copies. Our cell is going to get longer. It will start to pinch in, and eventually it'll pinch apart into two separate cells, each one having a set of genetic information. So just know that in a bacteria, when they need to divide, they undergo binary fission. I know that was a lot of stuff. I hope you followed along. My name is Mr. Kite. This is the Lab 207 webcast, and I will see you next time. Have a good day.